Hello all, this is a short intro that I'm putting ahead of the original intro of this video. The rigors and complexities of YouTube content creation. There are times when you shoot content and you record it, and then on the back end editing out processing phase, you see that you said things that you might want to clean up and you might want to revise, and this is particularly important for the sciences as accuracy and your accuracy is critical for your reputation and for whether or not your peers believe you. So throughout the video, I'm going to make this short. Throughout the video, I talk about the various protein levels, and I realized that I said a couple of times, just a couple, quaternary, and the term is actually quaternary. I referred to a professor in this video, and one of the things she said years ago is that as scientists, it's important for us to go back and to continue to relearn certain things. And once you don't look at something for years, sometimes the words get jumbled and you can insert a letter where they don't belong. So the term is quaternary and not quaternary, but I think it wasn't worth going back to reshoot, and it doesn't take away from the entire the messages in the video. The other thing is that I referred to the term tetramer, and I want to be clear that a tetramer doesn't need to or doesn't necessarily involve identical parts. As you'll see, I talk about 4 parts, but all the parts don't have to be identical. So I just wanted to say this really quickly. Accuracy is important when you're a scientist and when you're a professional, and these might all be separate videos in themselves, but I just wanted to put that before this one because these are important concepts scientifically. So enjoy. Proteins have primary, secondary, tertiary, and quaternary structures. Welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this video, please share it, and please subscribe to my channel. Well, first of all, I want to announce that Big Discussions 76, Science and Technology recently passed the 1,000 subscriber threshold. And some really good things are going to come with that, and I want to thank everyone who contributed to the success of the channel, and also helping to elevate the channel above the 1,000 subscriber threshold. And I look forward to what's to come in the future. This particular video is going to take us back into the realm of the biomedical sciences. As I described in my recent interview with Dr. Werner Morris, who has a background in chemistry and the atmospheric sciences, I want to open this channel wide up. I want to make it a STEM hub. So while my particular backgrounds are in pharmacology and toxicology and the biological sciences, I want to bring on other experts and guests in other aspects of the STEM acronym: science, technology, engineering, and mathematics. So if you're watching this and you're in one of those other areas and you want to come on and talk about what you know and what you're doing, please reach out to me. If you know someone who's doing exciting things, who might be interested in coming and discussing what they're doing and what they're an expert in, please reach out to me. And if there are other topic areas that you want to hear about, reach out to me and let me know, and I'll search my network and see what I can find, and I'll see if I can create content on it myself or bring those experts in. So I just wanted to say that before I jump into the subject matter of this video. I want to continue to build this channel, not just in my area of expertise, but I want to turn this into a STEM hub. So in terms of this video right here, I just want to say before I dive in to the meat and potatoes of this video, I just want to say that some things, there are things on this channel I talk about for general knowledge and education. Some things I want to relate back to what's happening in the world right now. As you know, we're going through this world event, and I made an attempt to relate the last two pre-recorded videos on the Western Blot and on mutations and variances. I tried to relate that to the world event, and I'm going to try to relate this video to that as well towards the end. There are some things that we're not allowed to talk about openly here on this platform, but as is the case when you do a live stream or when you shoot a pre-record, sometimes afterwards you think, okay, well, I should have said this and I left this out. And the topic of this video I felt like could have gone into the last pre-record, but when you leave something out, that leaves the opportunity for more content. The other thing I want to say before I jump in is that I want to challenge viewers to think about everything in totality. So I spend time here presenting some data. I know the scientist Supreme, she presents a lot of data. There's another channel run by Rodney. He is in the clinic. And I want to challenge viewers to look at everything in its totality. So some of the basic concepts I talk about here and some of the data, a lot of this, it goes together with the things the scientist Supreme talks about, and it goes together with things that are happening in the clinic. So don't just think about some of these basic scientific concepts and terms and principles I'm talking about here in isolation. Think about them in terms of everything that you see, and think about them in terms of other contexts: cancer, pharmacology, toxicology, medications, disease states, and sicknesses of all kinds, because understanding those larger fields and contexts are often, the understanding of those aspects are built upon these basic concepts. Okay, so proteins have primary, secondary, tertiary, and quaternary structures. I'm gonna make an attempt to move through this in a very efficient and expedient manner and not try to repeat things that I've said in earlier content. You can look through my catalog, for example, to see what transcription and translation are, and to get an understanding of what amino acids are and those types of things. But I wanted to have a short discussion on protein structure, and my first lessons about this actually came at Johnson C. Smith University right there, and I want to acknowledge Dr. Sutton. I remember reading about this in Concepts and Principles of Modern Biology, and this is one of the first concepts that she taught to us. So in terms of proteins, as I've said numerous times, proteins are the workhorses of our cells, whether those are enzymes, receptors, cytoskeletal proteins, transcription factors, and so on, et cetera, et cetera. I'm sure you've heard the scientist supreme talk about transcription factors in the context of cancer, but they're the workhorses of our cells. And as I discussed in the last video, the sequences for those workhorses come from our nucleic acids. Now, when those sequences are processed or translated, or should I say transcribed, to create RNA strands, and those RNA strands are translated to make the proteins. Those amino acids, we know that there are amino acids. Those are the precursors of the proteins. Those are the smallest units. Those amino acids are covalently bound into peptide bonds. So when you're down at the very, very basic level looking at the linkages of multiple amino acids, that is the primary structure of our proteins. Now, depending upon how those amino acids are strung together, and depending upon the identity and the properties of those amino acids, because we know that some amino acids are more polar, some are more nonpolar, some are more acidic, and some are more basic. Depending upon the identity of those amino acids in those chains are either going to form what we call alpha helices, similar to DNA helices, but in this instance, we're talking about proteins, or they're going to form in what we call beta pleated sheets or beta sheets. And when you look at ribbon diagrams, I've shown ribbon diagrams on this channel. Ribbon diagrams attempt to show three-dimensional structures of proteins, and if you look at some of them, you can see alpha helices in the structures, and you can see beta sheets in those structures as well. The beta sheets are usually flat structures, and the alpha helices are spirals, and those can be connected by straight loop proteins. But those are the secondary structures: alpha helices and beta pleated sheets. Now, when you think about the tertiary structure of a protein, we know that the amino acids are strung together or polymerized in peptide linkages. So you get these long chains of amino acids to create peptides and then polypeptides. And then depending upon the properties of those amino acids, they arrange themselves naturally in alpha helices or beta pleated sheets. So when you think about the tertiary structure, now you're thinking about how those two lower levels are coming together. I'll say coming together so that you get a three-dimensional structure. So I stated that different amino acids have varying properties. Some are polar, some are nonpolar, or some are hydrophilic and hydrophobic, which means some readily want to interact with an aqueous or a water-like solution. Some are hydrophobic, which means they don't want to interact with those types of environments. Some are acidic, some are basic, and so on, et cetera, et cetera. So once those, depending upon how those are strung together and depending upon the sequence, you're going to get a three-dimensional structure where those organically fold into a conformation that's unique to that particular protein. So again, I want to draw your attention back to the properties of those individual amino acids. In the previous video, or maybe it was my Western blot video where I gave a more detailed discussion of the procedure, there's an amino acid called a cysteine, which has a sulfhydryl group. Well, nature is very smart, and what happens is for a given protein, those sulfhydryl groups are in sequence in specific parts of the protein. So when that protein goes to fold in its three-dimensional conformation, those sulfhydryl groups, those cysteines, find themselves in proximity, and you can get a sulfhydryl linkage. In some instances, again, some of the amino acids are polar, so you can get hydrogen bonding between different parts of the that protein. That helps it form its three-dimensional conformation. And then also, as I stated before, some amino acids are hydrophilic and some are hydrophobic. So those hydrophobic amino acids, those ranges of the structure, oftentimes will organically orient themselves towards the interior, while the hydrophilic amino acid regions will orient themselves to the periphery of the protein. So I'm using my hands here to try to show you how something forms and folds into a three-dimensional structure. But that's the tertiary structure. You want to think about a three-dimensional conformation that is dictated by the chemical properties of the amino acids in that entire protein sequence. Now, when you think about the quaternary structure of a protein, you want to think about the fully developed, fully equipped, functional, active protein. So, for example, I just described the tertiary structure, which involves the three-dimensional folding of a protein that's been translated. So in this context, let's just say that that's a monomer. So that means that's one portion or one subunit of an entire protein. I'm going to use the example of oxyhemoglobin, which is the protein that carries oxygen from our lungs to our extremities and carries carbon dioxide back to our lungs for so we can respire it and exhale it out into the atmosphere. There's oxyhemoglobin and there's methemoglobin. But hemoglobin itself, hemoglobin is a tetramer, which means that it has 4 parts. So, and it's not just 4 identical protein parts or monomers, but you also have the heme groups in there as well. So when you think about the quaternary structure of a protein, that's the entire structure of it. It's all of its parts. For hemoglobin, it's a tetramer, so there are 4. But it also has its binding regions. It also has the heme in its pockets, and all of its proteins are locked, and they're all associating together. And as I described in my Western blot video, that's its natural state, and you can denature that any protein with reducing agents like SDS, sodium dietyl sulfate, or thiothriotol or DTT, and then vortexing as well. So that's a protein's quaternary structure. So you might be saying, Dr. Dunbar, what does this have to do with our current world event and the medications being prescribed and are highly recommended for our current world event? Well, when you think about a contagion of any kind, your body is going to create antibodies to assist you in fighting off that infection. And for the current medication being prescribed and highly recommended for this current world event, that medication is basically boosting your immune system and boosting your antibodies to recognize this current contagion or a specific part of this current contagion. In the last 2 videos, I described and introduced the term epitope, which is basically a region of a given protein that an antibody recognizes and grabs onto. So in the last video regarding mutations and variances and those kinds of things, I suggested that if a specific epitope changes, then the antibody's not going to be able to find that particular contagion. And then afterwards I realized, you know what? It may not be that particular epitope changing, but if another sequence of that contagion or the part of that contagion changes, you could also get a conformational change in that particular protein, so that that epitope that was once available, that epitope might be buried within that new structure due to the mutation and the variance. I hope I didn't lose anybody there. Okay, so let me go back and do that again. So you could get resistance to the medication because the epitope that the antibody once recognized has changed. So the antibody is looking around and it can't find that epitope because the epitope changed. But you could, in theory, also get a mutation causing the structure of the protein to change, so that that epitope is now buried within that protein structure, and now the epitope, and now the antibody once again can't find it. And you, of course, you can also get both, where the epitope has changed and also it's become buried within a new protein conformation because there's been some sort of mutation to the nucleic acid sequence. So that's just, I'm speaking in hypotheticals here, but I thought that this would be a good way to have a discussion and educate those unfamiliar with it about how these particular things can be happening, because we're in this world event, and one of the concerns with prescribing the type of medication that we're using in this world event, a concern is that that medication may become obsolete and may no longer work after a while against this particular contagion, and there are reasons why. And I think that I hope that I've said something meaningful and added to our body of knowledge about why that might happen. So I'm going to stop this video here. Hopefully I didn't leave anything valuable out. If you have any questions or comments below, please let me know. As I stated at the beginning of this video, Big Discussions 76 Science and Technology recently crossed the 1,000 subscriber mark. So right now I am waiting for my community tab to boot up, and I would encourage viewers to keep your eyes on the community tab, because once that goes active, I'll be able to share articles and other links to inform you guys, but also I'll be able to share things that I might not be able to verbally speak and talk about, if you know what I mean. So I'm going to stop this here. Please like this video, please share it, and please subscribe to my channel. If you want to throw something in the tip jar, that information is below this video as well. Take care, guys. Stay safe and be safe. And as always, remember that your attitude determines your altitude. Take care, and I'll talk to you the next time. Bye-bye.